The next question is, explain how genetic modification can be used to change the function of an enzyme so that it acts on a different substrate. This topic relates to the genetic engineering chapter, which we will study later in the course. Since it's a detailed 5-mark question, we'll provide a detailed 5-mark answer, so we'll write. Genetic modification is a change in genetic material. Enzymes are proteins, and a gene codes for a protein or enzyme. By changing the gene through genetic modification, a new sequence of bases is introduced. This leads to a change in the sequence of amino acids, which causes a change in the shape of the enzyme molecule, especially the active site. The active site can then have a complementary shape that fits a new substrate, allowing the enzyme to act on a different substrate. Now we have another paper 1 question. The graph shows the effect of substrate concentration on the rate of an enzyme-controlled reaction. The results have been obtained using a fixed concentration of enzyme at a temperature of 30 degrees Celsius. Which statement explains the shape of the graph from X to Y? Out of all the given options, we would choose option B, the enzyme active sites are not all occupied. This is the correct answer because between points X and Y, the substrate concentration is increasing. At this stage, not all of the enzyme's active sites are occupied, so more substrate means more frequent collisions between enzyme and substrate. This leads to the formation of more enzyme-substrate complexes, resulting in an increased reaction rate. Then, the next question asks, which statement describes enzyme-catalyzed reactions? Out of all the given options, option C is the correct answer. We've likely come across this question before. Option C is correct because increasing the temperature from 0 degrees Celsius to 20 degrees Celsius raises the kinetic energy of both enzyme and substrate molecules. As a result, they move faster and collide more often and with greater energy. This increases the number of successful collisions between the enzyme's active site and the substrate, which leads to a higher rate of reaction, up to the enzyme's optimum temperature. Also, just to clarify, this question is not from the May-June paper, but from the October-November 2023 series. That's the correct reference. Yay! Finally, there's an entire question dedicated to the chapter on enzymes. Until now, we've only encountered smaller parts of this topic within questions related to other chapters, so never mind. This question states, amylases are enzymes that are made in specific cells of animals and plants and also by bacteria. In humans, amylase is secreted into the alimentary canal in saliva and in pancreatic juice. Now, part 1 of the question asks us to explain what is meant by the term enzyme. So, we'll simply write the definition. An enzyme is a biological catalyst that speeds up chemical reactions without being changed itself. Moving on to the second part of the question, it asks us to outline 2 structural features of enzyme molecules. We know that enzyme molecules are made of proteins, which are long chains of amino acids. They also have an active site with a specific shape that is complementary to the substrate. For the third part, the question asks, name both the substrate and product of amylase. So we'll write, the substrate of amylase is starch, and the product is maltose. The next part of the question includes a bar chart and states, amylase produced by bacteria can be used as part of an industrial process that converts plants such as corn into biofuel. Biofuels can be used instead of diesel or petrol. The bar chart illustrates how the activity of amylase varies with changes in temperature and pH. Then, it asks us to describe what the data in the bar chart show about the activity of the enzyme and explain why this information is useful for the producers of biofuels. Basically, this question is asking you to interpret the data presented in the bar chart and then relate your findings to a practical, real-world application. Our response needs to be written in 2 parts. So, we will write, the data in the bar chart show that the enzyme is active between pH 6 and pH 8, with the optimum pH at 7.5. It is also active between temperatures of 30 degrees Celsius and 70 degrees Celsius, with the optimum temperature being 60 degrees Celsius. This information is useful for biofuel producers because it helps them to maintain the optimum conditions for enzyme activity. As a result, biofuel can be produced more rapidly, efficiently, and cost-effectively, with a maximum yield. Next, we have another question which states, the cell wall of a plant cell can be removed by treating the cell with a digestive enzyme. And we have to name the substrate for this enzyme. In this part, we're being asked to identify the substance that the enzyme acts upon to break down the plant cell wall. Since the cell wall is primarily composed of cellulose, and the enzyme breaks it down, the correct substrate to name here is simply cellulose. Then, the next part says, some plant cells from part B of the leaf cross-section were treated with this enzyme and then placed in distilled water on a microscope slide for 1 hour. The cells were clearly visible using a light microscope at the start of the hour. The cells were not clearly visible using the same light microscope at the end of the hour. So we have to explain changes to the structure of the cells that took place between these 2 observations. So, we'll write, water entered the cells down a water potential gradient by osmosis. This caused the vacuole to take in water, increasing the size and internal pressure of the cell. Since the cell wall had been removed, there was nothing to prevent the cell from bursting. Coming back to multiple choice questions, this one shows a graph of the rate of reaction versus temperature for an enzyme-controlled reaction. It asks, what does X represent in the graph? And if you look at the graph, you can see that X marks a decline in the rate of reaction. So what's happening here is that as the temperature continues to increase, the enzyme activity starts to drop, and that's because the enzyme is becoming denatured. So the correct answer here is option C. The enzyme is becoming denatured. Alright, let's look at the next question. It asks us, which of the following statements are correct? We're given 3 statements, and we need to decide which ones are true. So let's go through them one by one. Statement 1 says, enzymes are proteins and function as catalysts. That's true. Enzymes are biological catalysts made of protein. They speed up chemical reactions without being used up. Statement 2 says, enzymes are changed by the reaction they catalyze. This one is false. Enzymes do not get changed during the reaction. They help the reaction happen, but they stay the same at the end. Statement 3 says, the lock and key hypothesis explains the way that enzymes function. That's true as well. The lock and key model explains how the enzyme's active site has a specific shape that fits the substrate, just like a key fits into a lock. So, after checking all three, the correct answer is option C, because only statements 1 and 3 are correct. Alright, this is the same question we looked at earlier, so it's basically a repeated one. The same question, what's X representing here in the graph, and you can clearly see it shows a decline. So, we know that option C is the correct answer here, as it represents a decline in the rate of reaction. As the temperature increases, the rate of reaction decreases because the enzyme is becoming denatured. So once again, option C is the correct choice.